I basically have set up a computational microbiology and informatics group in Nestle. And so today, though, I'm going to talk about a project that I've been running with NIAID funding. We're in the 10th year, and it's also under evaluation for competitive renewal for 5 more years. I've continued to work on this project even during my period at Nestle, and I think this project embodies essentially the kind of capabilities from bioinformatics that the food industry, clinical scientists, and others need in order to make use of the data for the kinds of questions that they're interested in. So just a quick understanding of the audience there. How many of you come from sort of life sciences backgrounds? If you could raise your hand. Okay, the vast majority, good. Anybody from computational sciences, mathematics, exact sciences, engineering kind of backgrounds? Okay, for those of you, this talk is not going to go in depth on those areas. I'll be happy to talk to you in depth about those after. And then how about psychosocial sciences? Anybody from psychosocial sciences? So I would say that that ends up being a very important thing. I won't talk about it today, but how people react under stress and pressure of things like an outbreak completely changes their behavior. And so systems have to also be such that they can adapt to that behavior. So I won't talk about that today. Anybody from the business world, MBAs kind of folks? Okay, so the data I'm going to show you here, of course, is public data. And so I'm not going to show you any Nestle data, and I'm not going to officially represent Nestle's views in any form or capacity here today. But I do know the use case for the food industry, having been there for some time building resources for them. Okay, I got 2 of these. Is there one that actually works? Yes. Okay. So the first question is, okay, what does the food industry in general want to know about an outbreak? And I would say that it's not that different than most epidemiological users might want. So basically they want to know, first of all, what organism is this, and then where did it come from? And here on the right you have just an example from the CDC, you know, the CDC website about some examples of foodborne outbreaks. I think for this crowd you don't need to hear that, but of course it's important. And people get concerned about, you know, bacteria in their products. So, oops, sorry. What is unresolved, though, is this bottom part, and we already had a question about that. What the definition of sameness, there is no single definition of sameness for all questions. It's a contextual, and it depends on your question. And so, for example, we've already heard, and you know, I'm going to date myself, we were designing and building our own PFGE instruments when I was a grad student because the technology, you couldn't buy one yet. And I did a lot of work in that technology, but it turns out that it could be that for many applications, the current gold standard of PFG is perfectly fine. If it answers your question appropriately, it's fine. And so sameness is, there is no unique and all question independent definition of sameness. If you're dealing with Bacillus anthracis in an outbreak like the anthrax letters, there's going to be a very different legal standard and all of definition of sameness than there might be if you're dealing with the food outbreak, and it also depends on the organism. Organisms evolve differently, and they may change and rearrange a lot more than others. And so once again, I think the critical point isn't to try to define sameness for everybody, it's to say for your specific case, what is good enough definition of sameness, and then define that as a standard operating procedure. And then you can go ahead, and you don't need to get in that argument anymore. The second point that's also very important to the food industry, but I would say that all clinical scientists I've worked with also feel the same way, it's the issue of confidentiality. And so, you know, people don't like to, for a variety of reasons, have data out there that, you know, may be used in some way to interpret that they haven't been doing a good job or whatever, things like that. And so this issue also comes up as very important, and I'm not going to resolve the issue for you today, but those are unresolved issues that must be implemented in any kind of solution. So over the many years, I've been privileged, you know, to participate in a large number of international meetings that have been discussing this issue of How do we move to genomic enabled outbreak detection and analysis? And I'm not going to go through the gory details here, but we've been fortunate because of those meetings to essentially come up with what I call the workflow, the use case that embodies this. And in terms of the PATRIC project, there are many of these that you see with this coloring that are already in place and available freely to the community. This is a NIAID-funded project. Some of them are planned still to be completed in the next few months because the current cycle ends in September. And then assuming that we make the competitive renewal for PATRIC 3, some of these other ones will be implemented in the very beginning of PATRIC 3 year 1, which starts in October. And then some will be developed much later, especially with respect to clinical aspects of interpretation and use of data. So that's the general workflow that has essentially come from interactions with the community at various meetings. And that's what's being implemented. So you've already heard about sequencing. I'm not going to go through that. But basically what most users need that want to do computational epidemiology in a genomic world is a place to upload the results of the sequencing, the raw reads. And then we would like to see this whole process fully automated. Now, every step of this process that isn't automated, that requires human intervention, that costs time and money. People are expensive, electrons are cheap. And so you may say, oh, well, I don't believe that that will ever be automated. Well, let me tell you that our previous speaker talked to you about some of the first bacterial genome sequence. It took many months to annotate those. Today, you can annotate a bacterial genome in minutes. And that's because it's been automated. And there have been a number of studies that show that on average, it's just as good as human curation for annotation. And that just turns out to be that over time, we've learned. So I feel like very rapidly we need to, because of the dwindling cost of sequencing, make it very easy to do all of this inside a computer. And so the first step, and I'm not going to go through all this, but I just want to give you an example of the complexity, is how do you take those raw reads, pre-process them, analyze them, assemble them, post-process them, and then assess the quality of that annotation. And you know, in each box, I'm giving you a number of algorithms that are out there that have been developed. So one approach would be we sit and argue for a long time about what are the best algorithms. Again, it's contextual. It just depends on the genomic architecture of the organism in question, the sequencing technology used, et cetera, et cetera, et cetera. So a computational approach, since electrons are cheap, is essentially to say, well, let's investigate all of this and let's build a system that eventually converges on the best possible assembly entirely through using all these different capabilities, okay? And then once you have that and once you understand that, you can now fully automate this process. So again, I'm not going to go through gory details here. For those of you, few in the audience that are computational scientists, I can give you more detail. But essentially what we've done is we've gone and looked at some data where different sequencing technologies are used. But again, we always start with the same DNA. And people have taken exactly the same DNA, sequenced them with different sequencing technology. Different assemblers are used, and so on and so forth. And then we look at things like compute time, like the quality of the assembly, based on a set of criteria. And what we find, of course, is that it depends on which organism you have and which sequencing technology you've used. But if you do this enough, what you can figure out is what is the right combination for each organism of interest. And if that's included, if the information about that is included in the metadata, which we'll talk more about in a minute during upload, then that can trigger the best assembly protocol in an automated way for that particular type of organism. So inside PATRIC, the advantages that you have is that all the genomes, this was the last count, but it's growing exponentially as you can see, are annotated to a single SOP, which makes comparative analyses trivial and actually highly enabled. If you have different protocols for doing annotation, you can argue about which one's best, but the point is then you are chasing artifacts of different annotation protocols rather than things that you might be interested in as an end user. And so I'm not going to go through the whole website or anything like that, but I just want to point out to this piece here. There is a personal workspace, and this starts to get into the sociology of the community. Most people are not willing to, in the process of utilizing their data, essentially release it immediately to the public. They want a place where they can work with it. So in the personal workspace, That's where you would do that. And it doesn't matter who you are, you could do it, and you could share it with only the people you want, including nobody, if that's what you want to do. And so the integration of a variety of genomics data and metadata, and as I say, we'll talk more about that because that is really key. You can think of genome sequencing as an assay, right? The real data are your metadata. The rest is just the result of an assay, even though those of us who work in genomics might be offended by that at times, but that's actually what it is. It could be any assay. It could be a metabolomics assay, expression assay. They're assays. The real strong data is the metadata, so we really need to work on that aspect. But you have it integrated here, and you can summarize it at various levels. So you see an example here where it's summarized by my favorite group of organism, the Rhizobialis. That includes, for those of you who are ID people, of course, Brucella. But basically here, anytime you're in a particular group, everything you see, including other types of data, is contextualized based on the group that you're in. And one of the just simple brain-dead things that you can do with metadata is say, well, I want to see, where all these isolates are coming from. And you can see, based on the isolation country, where they're coming from. And not surprisingly, a lot of the public data is coming from US-based isolates. Now, I want to really stress how you do this. And so the way we build PATRIC, and because I'm both an engineer, but then a microbial geneticist lab rat, I grew up in the lab doing pulse field gels and other molecular genetics type of work, is you actually have to sit and co-create and collaborate with the people who are actually trying to do something. The notion that we could abstract ourselves from that and somehow say, well, this is the way they should operate, and then build a bunch of tools, and then it's round things in square holes, doesn't work. I've actually learned the hard way that that's not the right way to build resources. So what you need is to find communities of practice that are interested in collaborating directly with you. And because of my own specific interests, one of the places we started was in Brucella. And so this was what the Brucella community was doing when they went from 3 or 4 genomes in the beginning of PATRIC, so that would have been 9 years ago, to essentially projects where hundreds of genomes were being sequenced at once. And so what they were doing, and when you have 3 or 4 genomes, maybe this isn't such a bad idea, essentially managing things inside their own Excel spreadsheets. So when we looked at that, said, that's kind of cool. What do you like about it? And we started asking questions and interacting with them and understanding what problem they were trying to solve and how they were going about doing it. So in doing that, we then said, well, if we made that thing look like this, would you feel any better? And this is still being rapid prototyping in Excel-like stuff, right? And they say, oh, wow, that's cool, because now we can focus on the patterns. I said, okay, bingo. So is this helping you? And how is it helping you? And what isn't it doing that you're in? And so on, right? So we're collaborating with them in real time during the project. So as we worked on that, we next then essentially went from that to a productized version integrated into the PATRIC system. And essentially, there's nothing outstanding about this. It's basically a 2D viewer. This is common in informatics, used in all sorts of other things that have nothing to do with genomics. In genomics, it's gotten the name of heat map for some reason, but that's basically the name. And then all of a sudden, you now have something that is fully integrated with all the data in the website, rather than a standalone Excel spreadsheet where all I have is what I have. And I can now not only look at the things I care about, but everything else that's in there, right? Because it's standardized, annotated, and I can do comparisons at any level that I'm interested in. And so that became the product that came out of that research collaboration. And then we said, well, by the way, if you do that, you can also look at other types of data like protein families, right? With the same product, which is the 2D viewer, right? And so this is an example where we understand very clearly what a community is doing, but we productize it inside the website as something that everybody can use with much broader application than intended by the original community. And that's the part that we bring to the table in the collaboration, because if they were doing it on their own, they would still be in their Excel spreadsheet. And I have to tell you that our Brucella collaborators are very heavy users of this and have been very, very happy with the kinds of things that they can do now inside the PATRIC website, and they are the co-creators of it. And then once you have that, you can actually say, well, I don't really want to see things in tables. I'm done with that. I want to start to understand physiology, and essentially the same back end, if you want to speak, components can feed different visualizations. And so here you have somebody that's more interested perhaps in microbial physiology, and they can see those same data as EC number functions. And we've found that most of our collaborators prefer KEGG maps as a viewer for physiological biochemical pathway sort of data. We've actually had to play around with that a few times and tried a few different solutions, but we've converged on this. So this then allows people to take the same data, view them as 2D, view them as biochemical pathways, et cetera, under their control. Now let's start to shift for a second and talk about perhaps the elephant in the room, but yet the most interesting part. And I have to tell you that we've engaged in a collaboration across all the NIAID-funded sequencing centers and all of the NIAID-funded bioinformatics resource centers, of which PATRIC is the bacterial one, but there are others. And it's been over a 2-year collaboration to essentially standardize metadata and nomenclature. Now, when we started doing this in PATRIC, we kind of had to be a little bit ahead of the curve because community-based efforts are very powerful because they standardize for whole communities of practice. But of course, since they move by consensus, they tend to move more slowly, and that's appropriate. However, we had users who needed things immediately, so initially we took and did as much as we could based on what we thought, and of course shared that also with the community that was trying to standardize. And so the initial metadata essentially release inside PATRIC included more than 60 fields that were standardized, and it would allow you to essentially, once we understood that, we could parse it from a variety of sources. Of course, we had to clean it up, so there's some manual steps. But then this is the results in a paper that's just about to come out of this 2-year-plus effort coordinated by NIAID that involved all of their sequencing centers and all of their bioinformatics centers. And essentially it has ended up, and I'm not going to go through the details with you here, but you can look at the paper, with a number of different sort of core attributes that have standard controlled vocabulary terminologies used, which makes that potentially interoperable with any other number of controlled vocabulary examples, such as what's in the NCBI Bio project repository. Okay, so the advantage of this is broad standardization for a community of practice that then allows you to compute across different resources using the same terminology. And this is very, very powerful, and we're very excited about this now. So again, I'm not going to read you the whole table, but the bottom line is there are project attributes, and those are essentially uploaded by the sequencing center because that has to do with stuff that's happening over there. Typically, people have samples and send them to sequencing centers, and so the sequencing center is responsible for this metadata. And then there are the sample attributes, which tends to be collected by investigators or clinicians that are essentially they know that those metadata fields, and so that would be their responsibility to add the metadata fields related to their samples. But it's all combined, and here's a semantic network that looks at the project metadata, and again, this is using open biological ontologies, the OBO Foundry terms, so it's in principle interoperable with any other group using these terms in any other field. And for the core sample, here it is, and we've done mappings of these to the large repositories, and we know how to map directly to them. So, the other part I want to tell you about, and of course we've learned this by participating in outbreak kind of activities, especially the E. coli one in Germany, where data was essentially provided in real time, and we use that as a use case for what the future will look like, where sequencing is happening in real time, and informatics resources have to be able to make that data available analytically in real time. And so what we found is that there are things that we ended up calling specialty genes, but basically these are antibiotic resistance genes, transposable elements, virulence factors, and so forth. And we decided with support of NIAID to essentially go about the process of manually curating this information into PATRIC with data from the literature. And so we can't, of course, because manual curation resources are extremely limited and dwindling. that we had to essentially operate on a series of organisms that NIAID considered high priority. And we've released these now, and we have now tens of thousands mapped, and you can sift and look at them by different types of evidence, cutoff scores for similarities, and scope. This is the curation process. This is a paper that's submitted. We hope it will be in press very soon. So this is a table from the paper. And essentially there is, for each and every gene, a manual step that involves a curator looking and tagging publication, and then essentially tagging that gene to the genome in which it was found. Now, there are cases, of course, in which somebody has studied a virulence factor and there is no genome. And so this is an interesting case, but we're not sure that anything that you might call a virulence factor in a different genome is going to have the same contextual representation that it does in the genome that it was studied. So we have to be very careful about how you leverage that across genomes. There are many ways to access this data. I'm showing the example of virulence factors, but basically you can get at it as data from organism and from global searches, Google-like searches. And then it is fully integrated across many different aspects of the database. So depending on where you are and what you're looking at, you can have that information now available to you once it's integrated into a resource like PATRIC. So this is, again, just some screenshots of the example. We're now showing examples mostly in Mycobacterium tuberculosum, because this is the community we're working with, much like the Brucella one was before, to essentially develop this capability, but then the capability is available to all organisms. And so basically here you see results. In this case, there's been a query that's essentially looking at Mycobacterium tuberculosis. And you have examples here. You could filter on additional metadata, okay, like an Amazon shopping kind of thing. And you could decide which ones you want to see. And then more importantly, down here, you get to see the assertion from the literature with the PubMed ID that was made to classify this as a virulence gene, so the evidence. And then you get to, as I said, see it and be aware of it in various different contexts on the website, depending on what you want to do. Maybe you're a researcher and you want to find out more about the function of this. And so being able to look at which pathways it's in, where it's expressed, and what other proteins it's related to, and so on and so forth, could be of high interest for an investigator. One of the things that a lot of people have really liked is the capability to look at that gene and all the transcriptomics experiments that it has been seen in as a way to help them understand its function. This is in addition to what you might do with similarity, is to say, well, when is it expressed? And that has been really an important hypothesis generation engine for many bench biologists. The last one is the SNP data, and again, we're using the mycobacterium community to do this and to co-create this functionality with. And essentially, the idea would be a fully automated SNP computational pipeline, as I described earlier, for all the other steps. And essentially, what we're looking at in collaboration with this community is the entire complement of drug resistance. So this is for antibiotic resistance, of course, for TB. And essentially, the good news here is that they're characterizing them quantitatively for each drug. And so what we're finding is, as I said in an earlier slide, is, we've got a lot of different workflows, and for MTB, there will be a workflow that's optimal. And so from then on, it will be fully automated. And in the beginning, we're experimenting with different results. So as I say, in the last step, it coalesces into a single automated pipeline. So in conclusion, what I wanted to tell you here today, and hopefully you've heard, but if not, I can certainly answer questions and clarify, is that I think we are definitely in a time where genomic epidemiology can be enabled and is being enabled, but it's basically through a couple or three very important aspects. The first one is you must have high quality and standardized data and metadata. I cannot overstress the importance of standardization here. It is critical. Now, you shouldn't be scared of standardization in the sense of, well, it's going to quash creativity and all. You can always in the future decide to re-standardize. Sanger was the standard, and then it became something else, and then it can become something else in the future. But at any point in time, if you don't have standard operating procedures, you can't actually get to meaningful results in real time. Fully automated pipelines that I've talked about here would allow right off the sequencer to have full-scale results within hours. okay? And then that would then completely change things. I think the second aspect, of course, is computational ecologies, and these must meet end user needs and questions. They are not about me having fun as a computational scientist doing cool stuff. I mean, I can do that anyway, but the point is, if we're trying to solve a problem, it must be about solution of that problem. And I believe that, you know, the interfaces that deliver data to the users must be fed to them in a cognitively appropriate way. When we worked with clinicians in hospital settings, they just wanted to get a yes-no answer with a probability score. They don't want to see genomic data. Okay? So you need, but you can do that once you have all the computational infrastructure in place. I think the needs are best addressed by these transdisciplinary teams that co-create. It's not about I do my lab work and then the bioinformatician comes and does what I tell him. If you were the right person to tell him what to do, you would not need him. So it's about a community of equals participating and co-creating. And then I think PATRIC is one example. It's certainly not the only effort, and it's certainly not the only example that is working on enabling computational microbiology for bacterial organisms. And so finally, my last slide is to say that, you know, this kind of effort involves large team, and I'm not even putting all the people we collaborate with, or the slide would be enormous. But basically, we have the VBI side, and our big collaborator, our largest collaborator is at the University of Chicago, Rick Stevens, my co-PI, and then this is a picture of many of the VBI people that have been involved. And of course, we thank funding from NIAID for, you know, close to 10 years now. Thank you very much.